Hello, my name is Chuck Harp. I'm the Research Associate for the C.P. Gillette Museum of Arthropod Diversity on the campus of Colorado State University, Fort Collins, Colorado. We successfully completed the previous NSF grant digitizing and imaging Lepidoptera from our collection under the LepNet program, where we had entered over 182,000 entries and imaged over 15,000 moths and butterflies. The following video will walk us through some of the steps we learned along the way to help other groups with the iDigBees grant, in which we are again participants. Best practices, step 1, preparations. A, sheets of labels should be made in advance. These are to be printed on stiff paper, preferably better quality to ensure longevity in the collection. An example sheet is shown. Each sheet yields 800 labels. Separate boxes or trays should be made for each student or volunteer. Each tray should have the essentials: scissors to cut labels, a small hand lens to read labels, a pencil or pen, forceps to safely remove labels, a pinning block to replace labels once they are input, a small unit tray to hold stop sign markers. The use of these stop sign markers will be explained later. Footnote: the stop sign is made with an insect pin and a hole punch from red paper. End footnote. C, it is helpful to have several pinning blocks so several specimens can be removed from the drawers and safely placed while entering data. D, white adhesive dots can be purchased online or from office supply stores. 3/4 inch labels work best as Avery 5408 or Office Depot 3/4 inch multi-purpose labels. Best practices, step 1A, preparations. Footnote, creating catalog numbers for your institution. When creating the sheet of 800 unique catalog numbers for use in databasing, it is best to use an Excel spreadsheet. In our case, we chose to use capital CSU_ENT as our institution label, followed by a 7-digit number starting with 0000001. For the entire document, choose top align, left align, and wrap text under the Home tab under Alignment. When you've typed your first number, and with the cursor in the bottom right corner, drag down to line 21. Select the entry on 21 and key Control X, deleting the box. Scroll up to line B1 in this case and paste. In this case would be CSU ENT 0000021. Drag this down again to 21 and repeat this until you've completed 20 columns, ending in the column T. By using Calibri font in 11 point, you should be able to print the entire sheet of 800 labels onto a single sheet of label paper. End footnote. Best practices, step 2, assessing insect drawers to be databased. Identify the drawers in the collection that can be databased immediately, not requiring further updates on taxonomy, and are fully sorted to genus or to species. These will be the first drawers that will be entered by the student volunteers and citizen scientists. When a drawer is deemed to be ready to hand off to be entered, place an adhesive white dot on the front of the drawer. When your volunteer selects that drawer, their initials are to be written on the face of that dot. When the drawer is completed, a line should be drawn through the initials, signifying the drawer is complete and ready to hand off to the imaging team. Footnote: if the drawer hasn't been completed in one sitting, a small stop sign is placed at the last insect keyed. letting the person know where to start on the next shift. Again, the stop sign is made with an insect pin with a hole punch from red paper. Best practices, step 3, recruiting volunteers. We were lucky to keep many of our volunteers after the LEPNet grant was completed in 2020. We continue to have them key lepidopterists for various ongoing projects while waiting for the iDigBees grant to be approved. New volunteers have come from our offering tours of the museum and recruiting from the school's zoology club, natural resources classes, and our entomology programs. We currently have 11 students and 5 community scientists trained to do the iDigBees data entry and/or imaging. Best practices, step 3A, recruiting volunteers. When we find students that seem interested in the museum or in insects in general, we let them know about all the possibilities in volunteering for the museum, either in databasing, imaging, or helping to curate drawers in preparation to database. We explain that no skills in identifications are required for these positions, just a good attention to detail, some basic computer skills, and some basic skills in handling pinned specimens. Most volunteers catch on with a 15-minute training exercise. Most volunteers will do 20 entries per hour or more, some faster than others. We have found that most students will gladly volunteer time between classes or in free time during museum hours without ever promising a paid position if one should occur. This is a good resume builder, and students appreciate knowing this and that their contributions do make a difference. We have had student volunteers that have databased in the university library or in the film department archives, so those coming from the biological sciences aren't the only resource stream. Be open to students that have been influenced by other students. Word of mouth is important. Only 2 students have come from our bio-ag department. Most have come from other disciplines on campus, i.e. the zoology department and environmental resources. Be open to their scheduling. Most good students have jobs, activities, and various class schedules to work around. Be flexible and show appreciation when they can work for you. Having a schedule of their expected work days and hours will help you organize your day. Our volunteers generally work 2 hours a week to 10 hours a week. Best practices, step 4, curation of drawers. If the collection is not curated with current taxonomy, someone will need to be working those drawers ahead of databasers, ensuring previously identified unit trays have current names using resources with correct taxonomy. This is a good time to upgrade any older unit trays and to replace older header labels with newer ones. Curation of insect drawers is not a part of the grant, as it is assumed that drawers are correct and current. Once the drawers have been updated, an adhesive white dot can be placed on the front of the drawer, ready to hand off to a volunteer to initial and start entering data. Footnote: we edit our replacement header labels using scientific names and the author and the year of publication using Garamond 9-point font. Header labels are pinned to the top left corner of the unit trays using short header label pins. Taxonomy follows as close to accepted current treatments as possible. Inexpensive sources for header label pins are Hobby Lobby or Joann Fabric, sold as sequin pins, sold in 2 different sizes in bulk quantities for around $2. Best practices, step 4, data entry. We physically enter all label data directly into the Scan Symbiota datasheets. This is accessed at scan-bugs.org. This video is not intended to do a thorough review on how to enter data, but will try to give tips on that we have found to make things easier to process these specimens. Once each specimen is keyed and saved, the labels are replaced on the pin and the collection number is pinned. the last label facing down so it can be seen easier later. Best practices, step 5, imaging. Once the imaging person receives the completed drawer, they will select specimens to be imaged. For the bees, 3 images for each specimen are required: a dorsal view, a side view, and one of the front, best showing the face of the bee. We have found that most smaller bees can be shot with a single image with a 100 millimeter macro at 1:2 magnification and at f8 for better resolution. Larger bees will still have to be stacked. Labels are removed, the images are taken, and are uploaded as attachments to the scan entry. Footnote: once a specimen has been imaged and the labels replaced, a single hole punch color dot is added at the last, showing that the specimen has been imaged. Once enough images are completed in the drawer, one more of the color dots are glued to the outside of the drawer next to the white label, signifying the drawer has been fully databased and imaged. Finally, best practices, step 6, posting of daily, weekly results. We found it important to post photos of our volunteers inside the museum for recognition of their accomplishments. We update our completed numbers of entries at least once a week, showing our progress toward the final goal. Volunteers appreciate knowing that their time and work is valued. We want to thank our volunteer group for helping with the LEPNet grant and continuing with the iDigBees grant. These are some of our volunteers for this period.